be able to describe the limb growth and development and the various clinical conditions that are related to them. At the end of the 4th week of development, the limb buds become visible as outpocketings from ventrolateral body wall. Now, while we were discussing the general anatomy, we became aware of the fact that in the embryo we replaced the term anterior with the term ventral. So if you have a close look at this diagram, you can easily see two limb buds. There are two pairs, but just because we are looking at the lateral view, we can see two limb buds, one for the fore or the upper limb and one for the hind or the lower limb. The forelimb appears first, followed by the hind limb, and there is a difference of 1 to 2 days in the appearance of both. So if you look clearly in the diagram, you can see that this is the limb bud. The part where it is attached to the embryo, you can see that this is the root or the origin of the limb bud. So this is the proximal part of the limb bud, and this part is the distal part of the limb bud. Let's now have a look at the structure of the limb bud. In this diagram, it is quite clear that the limb bud, it consists of a mesenchymal core which is lined by cuboidal cells. The mesenchymal core of the limb bud is derived from the parietal or somatic layer of the lateral plate mesoderm. And you already are done with the germ layer derivatives, so I'm not going to go into the details of the mesoderm derivatives, but just for a recall, you know that the lateral plate mesoderm will form the bones and the connective tissues of the limb. So the ectoderm that lines the mesenchymal core, as we have already said, is cuboidal. Ectoderm at the distal border of the limb thickens and forms the apical ectodermal ridge, which is quite visible in this diagram. This ridge exerts an inductive influence on adjacent mesenchyme, which is this part. So under the inductive influence of the apical ectodermal ridge, the adjacent mesenchyme becomes a population of undifferentiated and rapidly proliferating cells, and its name becomes the progress zone. So it is quite clear in this diagram that this apical ectodermal ridge is causing the mesenchymal cells to proliferate in a very rapid way. Now let's move on to what happens in a 6-week-old embryo. In 6-week-old embryos, the terminal portion of the limb bud, it becomes flattened. It is quite clear in diagram B that the terminal portion or the distal portion has become flattened, and it results in the formation of hand plate and foot plate. It is separated from the proximal, separated by a circular constriction. So this is the distal segment and this is the proximal segment, and the hand plate is separated from it by a circular constriction. Let's move to diagram C. A second constriction later develops and divides the proximal portion into two segments, and the main parts of the extremities can also be recognized. Let's now move on to the development of the digital rays and digits. Digital rays and digits, or in easy words, fingers and toes, are formed when cell death in the apical ectodermal ridge separates this ridge into 5 parts. Now it is quite clear in diagram A how this process is going to occur. Further formation of the digits depends on their continued outgrowth under the influence of the 5 segments of ridge ectoderm, condensation of the mesenchyme to form cartilaginous digital rays, and the death of intervening tissue between the rays. And all these 3 steps, the beginning of cell death, the formation of the digital primordia, and the continuation of the process are quite visible in diagram A, B and C. Till now we have been discussing how the external shape of the limbs is formed, but let's now move on to what happens inside the mesenchymal core. In a 6-week-old embryo, While the external shape is being established, the mesenchyme in the limb buds begins to condense, and these cells differentiate into chondrocytes. We know that the limbs develop by intracartilaginous or endochondral ossification. So by the 6th week of development, the first hyaline cartilage models foreshadowing the bones of the extremities are formed by these chondrocytes. Now it is here that we need to recall our knowledge of general anatomy. We know that for long bones, we have a primary center of ossification which appears in the diaphysis, and from here the ossification it progresses towards the end or the epiphysis of the long bones. At birth, the diaphysis it usually ossifies completely, but the epiphysis they still remain cartilaginous. The diaphysis and epiphysis are united to each other by epiphyseal plate, which is also known as the growth plate. And once the growth of the bone is complete, the epiphyseal plate it disappears. As for the primary centers of ossification, it is usually said that the long bones, all the long bones in the body, they have primary centers of ossification by 12th week of development. This diagram is also a further recall of the process that the mesenchyme it gives rise to chondrocytes. The chondrocytes they give rise to cartilaginous models of the bones, and the primary centers of ossification they appear in the diaphysis, and from here the ossification it progresses towards the epiphysis, and finally at birth we have a fully ossified diaphysis and unossified cartilaginous epiphysis with the secondary center of ossification, and the growth plate or the epiphyseal plate it separates the epiphysis from the ossified diaphysis. And because bones are being formed and the bones in the limbs they articulate with each other at joints, so the formation of joints is also an important topic that must be discussed. So synovial joints between the bones begins to form at the same time that the mesenchymal condensations initiate the process of forming cartilage. In the region between two chondrifying bones, an area that is known as the interzone is formed. It also consists of a large number of cells. The condensed mesenchyme in this area differentiates into dense fibrous tissue, which further results in formation of articular cartilage covering the ends of two adjacent bones, synovial membrane, joint capsule, menisci, and ligaments within the joint capsule. This process it occurs under the influence of the regulator WNT14.